Welcome everyone. I'm Ricarda Schöpp, Senior Executive for Chemspec Europe, and I'll be your moderator for today's webinar. Today's webinar is presented by Dr. Andrea Adamo, CEO and founder of Zaiput. Zaiput have been attending Chemspec Europe since 2016, and Dr. Adamo is well known to our educational program, having presented last year on site in Basel at our Pharma Lecture Theatre. Before we start, let me just go through some basic functions of the GoToWebinar control panel. By clicking on your red arrow, your control panel on your screen will open, and there you find a section to ask questions. Please feel free to submit your questions during the presentation, as well as at the dedicated Q&A session at the end of the presentation. However, I would like to make all attendees aware that this session is being recorded and published for marketing purposes at our Chemspec Europe channels. As for now, ladies and gentlemen, without further ado, a very warm welcome to Dr. Andrea Adamo.
Good morning, everyone, or good afternoon. This is Andrea Adamo. Ricarda, thank you so much for the nice introduction, and thank you to Chemspec for this opportunity to present, and thank you all that are attending for making the time to listen to our story and our journey. What I would like to do today is to share a little bit of our story and share with you some observations that we've been making based on the use that people have made of our technology and feedback we have received, because we think that this could be very interesting for the growth of the entire community. Chemspec has been kind enough to provide us the opportunity to ask some questions. I would encourage you to answer, because the results will be shared, and it's really a way to gauge for everybody where we are as a community in the context of batch, flow, and their transition. And I will argue that hybrid systems seem to be an interesting way, or a realistic way to go forward, at least in the near future. So I will start with, so regarding the questions, we have 2 questions at the beginning and 2 questions at the end of the presentation. So Ricardo, if you could go ahead with question number 1. Question number 1 is, what type of experience you have? Are you batch chemist, flow chemist, or you've done a little bit of both? You're both a batch and a flow chemist. So let's give some time for people to vote. And now I think, Ricarda, perhaps we can move to the second question.
Yeah, I'm going to close that one.
Okay, great. I can hear you.
Would you like me to share the results, or do we?
Yes, yes.
Yeah. Okay. So 73% both have experience with batch and flow chemistry.
That's very nice to hear. So Ricarda, can I go on with question number 2?
Yes.
Very well. So when you perform a liquid-liquid extraction, which one are the 2 most important aspects for you in your process design? Is it the selectivity, the yield of the liquid-liquid extraction, the speed of separation, capital cost of the equipment, or operating cost? Which 2 are the most relevant to you? Let's give a few seconds.
Yeah, 63% now, but we're still having votes coming in. Yep, 75. 82. All right, I'm going to close the poll. And let's have a look at our audience results. Okay, yeah, so the two most relevant aspects are the first two, selectivity and yield, very well followed by speed of separation, operating cost, and capital cost.
Very nice. So thank you so much to everyone for participating. So let me go on. So why focus on liquid-liquid extraction? Here we are showing data collected in the context of pharmaceutical activities that show that extraction is the most common post-reaction step. And the same is true also in the fine and specialty chemical space. So if extraction is so frequently used and pretty much ubiquitous, the question is there anything that we can improve to make it better or to make it more effective? And so our own personal journey as Zaiput started in the context of continuous manufacturing or flow chemistry. So in flow chemistry, the idea is to go from reagents to product in a continuously seamless process. So in this cartoon, the gray steps, the gray pieces represent reactive pieces. And what we have seen is that the reality is that people have a piecewise continuity where reactions are done in flow and workup, liquid-liquid extraction, is always often arranged in batch mode. So the question is, can you actually do better? Are there ways that you could leverage to go closer to the ideal paradigm to leverage more, in order to leverage more the advantages of a continuous manufacturing scheme? And so the answer that Zaiput has tried to provide in this context is to carry out continuous separation. So let me do a step back. When you do a liquid-liquid extraction, let's say in a separatory funnel, you shake the two phases for mass transfer, and then you wait for separation. The idea here is to do it in a continuous paradigm, shaking the two phases in a line. Here you would have in the example red, it's water with some food dye, and the clear phase is toluene. And after you achieved mass transfer in your line, with static mixers, perhaps if you scale up, then you use a continuous separation device in order to carry out the complete extraction, complete the process step. So how do we accomplish this? We leverage wetting properties of liquids onto a porous substrate. So if you were to look at one of our devices, inside there's a membrane, and once you present the two phases, the organic typically wets the membrane, which is most often PTFE, and the aqueous is naturally repelled. So think of it as the non-stick pan you have at home. You pour oil and the oil will spread, you pour water and the water beads up. So that's exactly what we are using over here. Then we establish a little bit of pressure across the membrane as to push all the organic. Now, if this pressure becomes too much, we will also push the aqueous. So in other words, there's a small operating window in the pressure domain where you can push all the organic without pushing any of the aqueous. So in other words, there's a small operating window in pressure where you can achieve complete and selective separation. In order to keep this pressure difference, we have on board of our device a high precision pressure controller. It's a mechanical device that at all times controls the pressure across the membrane so that you're always within separating conditions, whether you change mixtures or you change flow rates or you add other pieces of equipment downstream your separator. So what we are doing here is we are shifting from a gravity-based paradigm where typically people would settle the two phases that have been mixed up together onto a paradigm where separation is accomplished by leveraging surface forces. And so there's some implications. The first one is the ability to separate emulsions, or in general, even liquids with same density. So the cartoon explains graphically why this is possible. So let's say that you have a water in oil emulsion with water droplet dispersed in this oil matrix or organic matrix. if you have an hydrophobic membrane, all the organic will permeate, forcing the coalescence of the aqueous. In a similar way, if you have oil in water type of emulsion, as shown on the right, by using an hydrophilic membrane, now you can drain out all the aqueous and force the coalescence of the organic. In this way, you can get very, very high efficiency of separation in a single stage, where high is typically something in the range of 94 to 100%, depending on the scale, how fast you're flowing, and some details of related to the system. Now, this is the first and most important one because it allows this type of selectivity, but there's other aspects that are related to the change of paradigm. So another important one is the possibility of scaling up. So the reason this is important to mention is because surface forces are known to be forces that play a role only on a small length scale, yet we are able to scale up the device. The reason is quickly explained. So what we do is we create large sheets of membrane and spread the liquid in front so they can still leverage a local interaction in the proximity of the membrane. Continuous operation, as I already mentioned, separating liquid with identical density. The concept is actually general and can be used also for the separation of gases and liquids, where typically you would have the liquid being the permeating phase and gas the phase that is kept behind. From a process standpoint, the availability of the possibility of separating phases with a very small internal volume offers very interesting advantages, especially in certain scenarios. Typical scenarios would be cases where one of the species is unstable, decays quickly, a quick quench or extraction is needed, and this is the fastest possible way this can be accomplished, but also cases where you have a safety concern, whether that be high energy materials or toxic substances or high potency APIs. Then, of course, there's other features that would stem from the design of the device. I would focus on the chemical resistance and plug and play functionality, namely it's very easy to switch out. So what we have seen happening is the following. So we have seen first, we come from flow chemistry, as I mentioned. The technology was developed at the Massachusetts Institute of Technology originally for some research projects. And the first applications were in flow chemistry, which is the natural avenue. And so I will share a couple of examples in that domain. And then I want to move on to hybrid processes, which is the next type of applications that we've been seeing flourishing, I want to say, in the last year and a half or two. And then mention a few things about liquid-liquid extraction, which actually is a continuous process that has been run a lot in batch domain. So it's as if this hybrid process that I want to mention today have already been foreseen in the industry in terms of applications before all this actually started. So let me dive now into some examples. So in the context of flow chemistry, one example is the synthesis of fluoxetine, which was presented. The chemistry steps were presented before us, and it's 3 reactive steps, and workup in between them was done offline. The reason is that the first dibold reduction produces a salt, an aluminum salt, that is incompatible with the amination step, and so removal of the salt needs to be carried out. And then after the amination, it's important to get to remove all the water because that hinders the aromatic substitution. So the chemists that work with us on this project basically set up a flow scheme with workup to remove the aluminum salt and workup to remove the aqueous. So this is more specifically what ended up happening. So after the first dibold reduction, a first wash is carried out. One wash is not enough to remove all the aluminum salt, and so a second wash is cascaded in essentially leveraging the modularity of the device. Now the process mixture is ready for the amination step. And after that, there's another separation to get rid of the water phase and start preparing the stream for the final aromatic substitution. This was done in the context of academia, and was done actually, all the process was pressurized, run at 17 bar, because reaction 2 and 3 were run hot to push the reaction yield. So in this example, which still stands as one of the little bit more complex ones, essentially what we showed was multi-stage operation with multiple workup stages leveraging the modularity of the device. What we have seen in industrial application is that people are using this, people are deploying this type of schemes. Currently, it involves usually less spaces, but this is definitely the direction that seems industry is taking for flow chemistry applications. Another example I like this for the elegance of the solution, is a 3 stages acid-base extraction. So the chemist here is as a mixture of crude atropine that needs to be purified and is done through this acid-base extraction steps. So the advantage of running it in a flow scheme is that once the flow scheme has been arranged, the process chemist can quickly go through a variety of configurations by changing pH, adjusting conditions, and can quickly converge to an optimal solution. And once the process is developed, pretty much the same scheme is what can be moved to the piloting plant and production eventually. I have one more. This is drawn from the work of Professor Oliver Kappe from Graz. Him, but also there's been many other scholars exploring so-called forbidden chemistries. So, namely chemistries that people could not run because their reagents are aggressive, toxic, they present some level of danger. So this is an example of cyanogen bromide that is synthesized in situ and then is separated out in order to be deployed for some synthesis. Of course, I am biased towards this example because the lack of headspace, the continuous operation, the small internal volume, and indeed the chemical resistance are all key elements to ensure that the safety of the process is maintained at all times, because the amount of cyanogen bromide present in the system is really, really small in all conditions. And there have been plenty of examples in the literature, but also in industrial applications with phosgene and diazo compounds have also received a lot of attention. I have one last application in flow, which is gas-liquid separation. We have seen in the last, I want to say, year and a half or 2 years, an increase in the frequency of these applications. And there has been essentially 2 modes that have been used to deploy this type of approach. One is use a separator to remove a gas stream that is present with the liquid. And the gas could be either an unreacted reagent, say a case of an hydrogenation, for instance, or it could be a product of reaction. So by removing the gas continuously, you can actually keep the liquid line pressurized if you wish to do so, and hence cascade right away the next unit operation. The other type of application that we've seen is actually addition of gas on purpose in order to strip from the liquid a volatile component. So once you add gas, the volatile component will partition between the liquid and the gas. So upon removal of the gas, the volatile component is removed out. And all these schemes, of course, are modular, so they can be nested or cascaded to achieve the desired level of removal or specific targets of the process. So to summarize, in flow, people have used the technology chiefly for inline extraction, And actually, we've also seen use for analytical purposes so that you can separate out a stream before having it go through your inline PAT tool. The advantage is that by separating before the PAT tool, you retain the real-time information as opposed to recover the information with post-processing, because the two phases will give glitches in the signal of the PAT tool. So I hope I've managed to share the type of cases that we have encountered in the last few years and what we see being the trends of the field in this context. Now let's move to applications, what we call hybrid applications. So in order to get into this hybrid approach, we have to focus on go back to what I guess everybody knows. How do you do an extraction in batch, and what is it that could be identified as non-ideal? So the first observation that we've made is that if you have a batch reactor and you have a reaction followed by an extraction, only half of the reactor will be used because you need the space for the extraction solvent. So if you simply put it in numbers down in terms of productivity in your production plant, this is, for the sake of example, 1,000 liters. You are using only 500 liters for your chemistry because you need the space for the solvent. So you're cutting your productivity by a factor 2 just because you have an extraction to carry out. There can be other time wastes associated. Examples are the top phase cut or rag layer, which may take more time to settle, or the compromise between separation efficiency and mixing. So it is known that the more you stir, the faster is your mixing, but then your droplets become smaller and hence your separation time goes up. So there's always a trade-off that is being made over there. Other set of issues, multiple washes. Sometimes you need to carry out your extraction process multiple times in order to meet a quality aspect. This becomes more stringent perhaps in pharma, where the quality requirements may be tighter than other fields, but indeed in general, since steps are done sequentially, 2 extractions are twice the time and 3 extractions would be 3 times the time, so it becomes heavy on the time domain. Last but not least, emulsions, namely slow to settle system. If there's a modest change in your system, sometimes it leads to hard to settle conditions, or there's compromises with the mixing. So the question that we started asking ourselves, but also the questions that, in all fairness, the users came to us with or suggested was, is there a way to do better? And so if you approach this problem with, let's say, a flow type of mentality, what comes natural is actually the following. How about we run reactions in batch, especially if there is no drive to change them, if it is established reactions or reactions done for 30 years, or there's cost drivers that are not there to justify a redevelopment in flow, how about to run only the extraction in flow and then go back to subsequent batch operation? Now, there's a few things that actually you can leverage that may be very interesting. The first one is you can fill up your batch reactor completely. So you have an instantaneous doubling of productivity with information that you already have because you've been running your batch chemistry already for a long time. Your extraction solvent can be straight out from the container your supplier provided it. If this was a mixture, it could be prepared, it could be prepared also in a flow scheme. It means only adding 1 or 2 more dosing pumps to the scheme. So you pump out your liquids, you have an engineered area for mass transfer to reach equilibrium, and a large scale, I should say very large scale continuous separator, depending on what's your size, in order to carry out the separation. Now, pumps are well-known technology. Mass transfer engineered mixing is also well-known. I'm referring to either passive mixers of the type of static mixers, or there's also active mixers in the market. The key point I want to make here is that by using a continuous separation technology, you can actually decouple the mixing from the separation, because you can actually separate also emulsified systems. And so by decoupling them, you can push the mixing and make it very repeatable and very scalable in a way that is actually already known, just needs to be deployed. And so what we think here, what we have gathered is that this is actually a simple way to start leveraging better your batch assets and introducing flow concepts in the production floor to really share these ideas with a larger set of people within the organization. Now, one issue was the loss of productivity. The second one that I mentioned was the challenge of multiple extractions, which in batch are done in a sequential way in the time domain. In flow, they are also done sequentially, but in the notion of flow, actually, is a stream. It's, you know, they're cascaded in series. That's how we call them. So in the time domain, it's pretty much done at the same time. So the complexity of the time domain can be translated into a little bit more complexity into the so-called equipment space. All you need to add is another pump and another stage, essentially an extraction stage. And what I would add is that this setup actually can be mounted in a cart so that it can be deployed in different locations within the batch production floor. Last but not least, the challenge of the emulsions we already covered. There is no change between batch and flow. This is inherently addressed by a membrane-based type of separation scheme. So I would go back and make a note a second on scale. So what is also important is to look at the process time. So what we have seen in flow so far is that our, for the sake of complete information, most of our experience is in the pharma realm. I guess because of historical reasons, because our first application started in pharma. And in the context of pharma, this is, let's say, 1,000-liter reactor at the production scale. And so when people go to flow and they transition a process, or they make a process in flow, at production scale, you are at flow rates in the order of 1 liter, 2 liters per minute, perhaps. Because since you are running for extended period of time, in the course of 8, 16, or 24-hour shifts, you actually pile up 1 to 3 metric tons, give or take, of material that has been processed. So you still pile up a lot of material. When you think in a batch, these 1,000 liters, once the reaction is done, will be ready all at once, and you want to process them quickly. So we think that the junction point between a flow paradigm and a batch paradigm is in a flow scheme that has nominal larger flow rates than what you would typically do in a strictly flow scheme in order to merge the two worlds and avoid waiting 8 or 12 hours for a batch, and actually push the throughput so that you can do this in 1 or 2 hours. And so it's in this adjustment of scales that we see hybrid being able to be leveraged to the fullest of its capability. So with this, these are the key ideas. We have seen, as I said, a growing number of applications in hybrid schemes, starting from the pilot plant, where people will have 50 to 100 liter batches, and they want to be able to double their productivity and have more effective campaigns when they run them on a pilot plant. I want to close with some observations and thoughts on multi-stage extraction. And I guess I want to focus on a counter-current version. So let me start with some background on that, and then I'll explain how I see this coming together as a kind of Unitarian thought. So when do people use multi-stage extraction? It's very well known to everybody that when you run an extraction, your solute will partition between the two phases, and the partition coefficient is the ratio between the concentrations of your solute between two phases. If your solute partitions evenly, you have a partition coefficient of 1 and an extraction efficiency basically of 50% because you remove 50%. So if your partition coefficient is low, in order to recover more material, you have to cascade extraction steps. The other scenario where that might be something to consider is when you have two solutes that partition, and they partition in a similar way. So we look at the ratio of their partition coefficients, and what we want to do is enrich our solvent with one of the solutes with respect to the other. So in this case, we're looking at the ratio, and if the ratio is close to 1, we need to cascade extraction steps. Now, people have known this for several decades. What they've also known for several decades is the idea to run this in countercurrent. Why countercurrent and what is it? So I have a schematic in the next slide. The idea is to recycle your solvent in further extraction steps. And the reason you want to do that is because if you make a simple projection of recovery of material, let's say with 1-to-1 phase ratio and partition coefficient of 1, in one stage you recover 50%. But if you take exactly the same amount of material, but you run it in countercurrent with 5 stages, you would recover 83% of material. And in 2 stages, it would be about 67%. So by changing essentially the hardware you're using and not the material that you have, not adding a single drop, not removing a single drop, you can actually capture way lot. So again, this is an old technique. What we saw is interest in resurrecting it to new use, so to speak, in order to leverage better the efficiency that it can provide. And so what we went on was develop systems of that kind for ourselves in order to test them. And this elucidates the countercurrent scheme. So for the sake of argument, here there is a target chemical inorganic, and it's traveling from left to right. And we would add an aqueous solvent for extraction of the target at the last stage. Will come in the fresh solvent, and then the aqueous will be recycled upstream and upstream again, and so on and so forth. And so this is essentially countercurrent, and we went on for ourselves to develop it at the laboratory scale in order to carry out experiments and, of course, test the scale up, because any solution should be, it's interesting when it can be scaled up, and developed, tested it in a variety of conditions. We did a lot of tests. I will mention only 2 here for the sake of brevity at this point. This example was developed with Novartis. Some of it was done when the technology was still at MIT, and Novartis had a collaboration with MIT and got interested, and eventually actually this example is published. So they gave us a case that where there's a complex feed with THF, ethyl acetate, water, and a bunch of alcohols. Water and organic are added for the sake of collecting an organic stream that contains alcohol, and I apologize for the error here. The aqueous would collect all the alcohols. So we use the platform as a process development tool. So for assigned conditions of flow rate, you collect your samples. We did it studying the role of stage number also, and by doing analytical work, you can see the trends that I showed you earlier on. And so it becomes an effective stage, an effective way to carry out development. In this case, my understanding is that Novartis is running this process somewhere with an extraction column. We did the testing independent of that, of course, but with different technology, but the fundamental process is still the same. It's a liquid-liquid extraction in We did some more studies. This is a case of an oxidation of alcohols in flow. We did it using commercial reactors, and it's a biphasic reaction. There was a first separation done to separate the two streams, and then a 3 stages counter current to recover the phase transfer catalyst. In this case, we had a little difficult separation. The density difference between the two phases was very small, and so it needed about a day to be completed in batch, and it was readily done. And we did this in the laboratory scale with the Corning reactors in this case, and also the pilot plant scale. So this takes me close to the end of what I had prepared to share with you. I guess there's a couple of thoughts more I want to share. The first one is by looking at a comparison table, let's look at the technologies before getting into the features. So people have done multi-stage extraction with batch settler type of systems. So essentially you use a batch and then you move it to another tank that is another batch where you don't stir, and so phases can settle. There could be slight differences in the way this can be implemented. Very simple to operate, very large internal volume just by nature of the design. Centrifugal extractors have been around for a little over 100 years, and typically they have been the way to go for cases where density difference is very small. They have a lot of moving parts. There's some upkeep that they require. Extraction columns is essentially a gravity-based system. The heavy phase goes from the top and travels down. The light phase travels from the bottom up. And there's different ways that plates shaking or rotating so that phases are mixed up, shaken together in order to enhance mass transfer. So counter current extraction is inherently a flow scheme that has been often deployed when you need efficiency of extraction, even in batch cases. Perhaps not as much the columns, but the idea of marrying batch world and continuous world has actually been already there. What we see in our own experience and journey is a revival of this idea, because it seems that people want to leverage the best tool for the application and find a way in a shelf full of tools, whether you call them batch or flow, to pick what is the most suited one for the process and the constraints that are on the table. Now, to stay specific in our domain, that is extraction, there's a long list of parameters that people could look at the pros and cons on different technologies. And the table does not want to cover everything. There can be differences of opinions also based on what experience. We have tried to compile this to the most accurate possible way based on feedback we have received. I want to narrow down the field to about 3 or 4 items that seem to be of most relevance in industrial segments where molecules have a high added value. Starting from internal volume, because expensive material, everybody wants to save expensive material, and the internal volume of the device scales with the amount of material needed to collect a data point, because it scales with steady state, with time to steady state. So starting at the laboratory scale, we are actually, the technology that we've been working on provides significant advantages, and also in the context of ease of scale up and separation with similar densities. Of course, for the sake of intellectual honesty, there's a couple of things I want to point out. One, of course, is that I may have a personal bias because we've been spent a lot of time working on this, and the other one would be the presence of solids. So a membrane-based phenomenon, membrane-based process may run into issues. There's a lot of ways to address that. It's just important to spend some time to look at them in detail. With this said, that takes me to the end, which is before taking your questions, and question number 3 and 4. So Ricarda, if that is possible, we can start with question number 3, that is, for what application would you consider applying a hybrid, applying this type of technology? You know, the possibilities would be enhancing productivity of a batch reactor. So by this technology, I refer, you know, to membrane-based continuous separation. Would you consider it for speeding up separation time, separation of emulsions, for strictly flow chemistry applications, or actually for none of the above? So we give some time for to people to cast their vote. Ricardo, are you there?
Yes, I'm here. We are now at 76%. I would close the poll now. If anyone else would like to submit their poll, please submit by now. Right, yeah, there are a few more coming in. Okay, I'm going to close the poll now, and let's have a look how our audience voted. There we go. So yeah, 81% of our audience today would apply this technology for flow chemistry, and followed by 60% speeding up separation time, and then enhancing productivity of a batch reactor, and separating emulsions.
Very good. Thank you, Ricardo. So then I guess we can go to the last question that we have. How likely are you to explore hybrid batch flow schemes? Very likely, likely, somewhat likely, or unlikely?
Okay, just a few more seconds. Okay, I would suggest we are closing the poll. Thank you very much for submitting, and let's have a look. Yeah, we've got in total 70% very likely or likely to explore hybrid batch flow schemes. Great.
Thank you. Thank you to everyone. So this takes me to the end of what I had prepared to share. I hope I have managed to share some of the things that we've been gathering through our journey, the enthusiasm that we have in the different applications that we have seen flourishing. And one of the intents with the, with the generosity and the availability of Chemspec was really bring these ideas to the table for the benefit of the entire community, to see in which direction this community can and should grow. And also I would point, all of us have been affected by the recent events in the pandemic. It's something that is shaking the platform, the planet. And the chemical industry, perhaps a little bit more pharma, does have a role in addressing what we are going through, whether it's now or in the next few months. So I think this is a compelling time for all of us. With this said, I will be happy to take your question, should you have any. Also, I would encourage you, we have some limitations of time, to reach out either through Chemspec or to me directly via LinkedIn, or just at my email address, with question, curiosity, or thoughts you might have. We think that dialogue within the community is the only way to grow for all of us. Thanks again for your kind attention, and Ricarda, please take over.
Yes, thank you, Dr. Adamo. That was a very informative insight. We've got at this stage no questions submitted, so I believe if there are questions, dear ladies and gentlemen in the audience, please feel free, as Dr. Adamo just mentioned, to get in touch with him via the email address as shown on the screen right now. And of course, we as a Chemspec Europe team are always there and happy to answer any questions. And we very much look forward seeing you at our next webinar, which will be updated on our website during the Digital Hub, and of course welcoming you as well on site at the next Chemspec Europe. So for now, thank you a lot, Dr. Adamo, for taking the time and for the webinar. And ladies and gentlemen, thank you very much for joining us today. I wish you all a very nice day and see you next time.
Very well. Thank you, Ricardo. Thank you all.